Hello, everyone. This is Dr. Ripley, and we're going to talk about the type of analysis called analysis of variance. And this is a little long to say, and so by convention, we usually say ANOVA or ANOVA, and that's the abbreviation for analysis of variance, sort of. It's first capital A, the O, the of, and the VA of variance. All right, so this is going to be a quick introduction to how you go about doing the simplest version of ANOVA, one-way ANOVA. It's called one-way ANOVA because you only have one categorical explanatory variable. So whatever treatments, different levels of the treatments you've exposed your different groups to, you're looking for an effect of that one kind of treatment on your response variable. And you're interested in comparing the means of those groups, and you have more than two groups, right? If you only have two groups, you can use t-test. And in one-way ANOVA, we're going to have independent groups. So this is really the equivalent of the two independent samples t-test experimental design, but we're extending it so that we can compare more than two groups at once. All right. If you do ANOVA on the same data you do two independent samples t-test for, you get the same result. But you can apply it to many more than two means. And the way that we're going to be able to do that is by looking at the ratio of the variance within groups to the variance between groups. All right, this probably doesn't make any sense to you right now, but we'll spend a little time on what that means because it's important to understanding what we're doing with the overall analysis of variance approach. All right, now remember, variances are things that we calculate when we're on our way to calculating the standard deviation. So we find this quantity and then we have to take the square root of it, and that gives us the standard deviation. The variance is that thing. All right. So if you think about, you already know what the standard deviation is, this variance that we're talking about, remember, is the square of the square of the standard deviation. And because the standard deviation is the square root of something, it has mathematical properties that do not make it tractable for the kinds of calculations we're going to be able to do with the variance, which is why we're using the variances. All right, so when we talk about groups here, those are each of our individual groups of individuals that are in a particular treatment. So in our two independent samples t-test setting, you can think of those two independent samples. Those are the same thing as groups. All right. So each group of individuals that we expose the same treatment to. All right. And each of the groups are independent from one another. All right, so as we are going through the steps of doing our ANOVA, we're going to summarize the results into an ANOVA table. This isn't just a convenient calculation step. This is how we're going to show the results of our analysis in our publication. All right, so this is a formal summary of the results of your ANOVA, and if you look at the table, the structure of the table is going to tell you what the experimental design is. So these aren't just a convenient summary table, they convey a lot of information to the reader. We're going to calculate a test statistic for all of the ANOVA type of analyses called capital F. And there is an F distribution, which fits to ratios of variances. So it's a distribution that is going to fit to what we're actually looking at as our test statistic here. And there are several assumptions, just like for t-tests, that the data, our response variable, has a normal distribution. that the standard deviations of the groups are equal or approximately equal. And an analysis of variance works best if the sample sizes are equal, or if they're unequal, that there's at least 5 individuals in each group. So we're going to have the most powerful experimental design if our standard deviations are equal and our sample sizes are equal. But sophisticated software packages will compensate for those. All right, so this figure is showing 3 different panels, and it's showing box plots of what the results of our experiments might look like. And this slide is intended to convey to you the idea of the variance within the groups compared to the variance between the groups. All right, so across the x-axis on all 3 of these panels, we have 3 different levels of our treatment. Those are our groups. So this would be how we'd show the results of the experiment. We've got box plots here because it's nice to show the variation around the means and how different the means are, but these could easily be plots where we just have the mean and then the error bars with the standard deviations. So in this first panel, the medians or means are a little bit different from one another, and the variation around the means is fairly small. In the second panel, we have the same amount of differences between the means as in the first panel, but we have really big variations around those means, right? So the difference in these 2 panels is a difference in the variances within the groups, all right? So the first panel has small variances within the groups. That means each group has a small standard deviation, whereas in the second panel, the groups each have large variance within groups. So lots of variance between individuals. All right. We don't have a big difference between the groups, right? Big difference between the groups is what the third panel is showing. So on this version of the graph, one of the means is for medians is all the way down here at about 85, whatever these units are. The second median is at about 120, and the third median is at about 160. All right, whereas on the other 2 panels, they're all between 110 and 140. All right, so when we say variance between groups, we mean how big a difference there is between the means of the different groups. Right. So in which of these panels might we want to conclude that there was a significant difference between the means? Right, so the one on the far right, we have the biggest difference between the means. So looking at it compared to the other panels, we might want to say, oh wow, those means look pretty different from one another. All right, it's going to depend. So what we want is a way to measure how big those differences between the means are compared to how variable each group is, right? Because if we look at the panels, the first and second panels, even though the means are actually the same, if we had big error bars in like in panel B here, we wouldn't really look at that and say, oh wow, those means are different from one another. Okay, but when we have small error bars around the means, even when the means are not as different from one another, we might be more willing to say, oh, you know, those distributions don't overlap very much. We want to we might be able to say that those means are significantly different from one another. Okay. All right. So what we want to be able to do is distinguish between um, how sort of we want to be able to evaluate how differences the means are from one another compared to the differences within each group. All right, so this figure is showing if we show them all three groups together on one histogram, that the between group variation is going to measure the difference between the group means, one compared to each other. And we're going to call that the calculation of this value that we get, the variance between groups. Now, the within group variation is going to be like the standard deviation of each of these little histograms or distributions, right? So we're going to get a large value of f when the variance between the groups is comparatively large compared to the variance within the groups, right? So it's always relative. It's always relative. There isn't one set value that we're going to say, oh, this is too much difference or not enough difference between the means, right? It's always going to depend on how much each of the groups varies within itself. All right, anyway, so this is going to be this value f that we're going to calculate. We're going to go through a set of calculations in order to find it. And as we are making that calculation, we're going to be summarizing and progressing through a set of calculations in this ANOVA table. So ANOVA table is going to have source of variation, right? Where is our variation coming from? We're going to have part of it that's going to be made up of between the groups, part of it that's going to be made up within the groups, and we're going to call that error. Sometimes this is listed as between and versus within. And then the total error or the total variation in the data set is going to actually be a sum of the variation between the groups and within the groups. Where this says total, it really is a total. We're going to add up the values in the row for the between groups and in the row for the error, and that's going to give us the total for this bottom row. All right. So first we're going to calculate the sum of squares. We're going to write down the degrees of freedom. We're going to calculate something called the mean squares. And then the F value is going to be the ratio of the mean square between divided by the mean square error. All right, so our variance of between compared to within, this F value, is going to be actually the ratio of these mean squares. So if we think about the equation for standard deviation, it's the root mean square error. All right. So we want the variance, so we're not going to take the square root of it. So we're going to get the mean square errors, right? So our mean squares, that's what we're calculating here. We're going to use those in the ratio. And in order to get the mean squares, what do we do when we calculate a mean? Well, we're going to add up the values, and then we're going to divide by the degrees of freedom. All right, so the sum of the squares calculations are equivalent to adding up the square deviations in our standard deviation equation. And then the degrees of freedom, we're going to divide the sum of the squares by the degrees of freedom. And that's going to be how we mean the squares. And so that's how this, what's in this table, right? So the degrees of freedom, we're going to know that from our experimental design. So the degrees of freedom between groups is going to be little k, which is the number of groups that we have, minus 1. All right, so if we have an experiment with 3 different treatments, a control, and 2 levels, little k is going to be 3, degrees of freedom is going to be 2. Just like degrees of freedom typically works. Degrees of freedom for our error is going to be capital N, which is the total number of individuals in our entire experiment. Right? If we think about each group as having a number of individuals in it, little n, our capital N is going to be the sum of all the little n's. Right? So 7 individuals in one treatment and 10 in another, and 10 more in the next one. We're going to have a capital N of 10 plus 7 plus 10, or 27. Right, so the degrees of freedom of error or within is going to be the total sample size for the whole experiment minus the number of treatments. And then the degrees of freedom total for the entire experiment is going to be capital N minus 1. Right, so if you actually add up k minus 1 and n minus k, you really do get n minus 1 as the sum. Right, so total means total. These equations for sum of the squares between, notice we're taking a deviation here. We're taking the difference between the mean of all the mean of the means, the grand mean here, and the group means. All right, we're going to multiply it times the little n in that group. We're going to first we're going to square those deviations. We're going to multiply times the little n for that group, and we're going to add those up. So we're going to have as many terms for the between groups sum of squares as we have little k's. Right. Sum of the squares error is basically we're calculating the error like we would in a standard deviation. Each observation subtracted from mean, and we're going to square that just like we would in a standard deviation. However, we're going to do that for each group, and then we're going to add those up across all k groups. Right. Equations look intimidating, but they're really just different versions of finding the standard deviation. All right, now there is a formula for finding this. We can find it individually, or we can find it by adding together the sum of the squares between and the sum of the squares error or within. All right, so once we have the sums of the squares, we have the degrees of freedom, we need to find the mean squares. All right, so we're just going to go across this table to do that. So we're going to take the sum of the squares divided by its degrees of freedom, and that's going to give us the mean squares between. We're going to take the sum of the squares error or sum of the squares within, same thing. We're going to divide that by its degrees of freedom, and that's going to give us the mean square error or the mean square for the within. And then f is going to be the mean square between divided by the mean square within, just like we had in words at the beginning, because remember, these are variances. All right, this value of f, once we've found it, we're going to use the f distribution in order to find the p-value. So the f distribution is going to look a little like the chi-square distribution, right? It's not symmetrical like the normal curve. It is right skewed, and it has degrees of freedom because it has two component parts that you're using for this ratio. It has the degrees of freedom for the numerator, and it has the degrees of freedom for the denominator. So when I say the F distribution shape depends on two kinds of degrees of freedom, we have to take into account the degrees of freedom in the numerator and in the denominator. All right, so in the numerator of what? In the denominator of what? All right, so we'll go back to the previous slide and look at our one-way ANOVA table. All right, when we calculate this F value, it's a ratio. It's got mean square B divided by mean square E. So the degrees of freedom of the numerator is just the degrees of freedom that we have here in the between groups row. All right, and this is typically going to be a small number. You aren't going to have 1,000 different treatments. You're going to have 3 or 4 or 5 or 6. All right, so we have a fairly small number for the degrees of freedom between. And then the denominator is the mean square error. So the degrees of freedom for the denominator, we've got right here in our ANOVA table. So if you get to that F table and you think degrees of freedom, numerator, denominator, wait a sec, where do I find those? It's all in your ANOVA table. So go back to that ANOVA table. All right. And this is some examples of how the values of the degrees of freedom in the numerator and the denominator are going to change the shape of the F distribution. So we can see where we have the biggest number of degrees of freedom, both in numerator and denominator. We have a somewhat less right skewed distribution. So as our degrees of freedom gets larger, we're going to see that this distribution sort of gets squished towards the right. Right, and on this figure, it's using little n and little m to represent what you thought might be numerator and denominator. But they are also referred to with the Greek letters nu for the numerator and nu and mu for the denominator. All right, so this is where those some of those different letters are used to refer to them. All right, so once we've calculated our F, we're going to use the F distribution table to find the p-value. Right now we've got degrees of freedom in the numerator, and we've got degrees of freedom in the denominator. So this table is the most complicated one. So we're trying to keep track of two different degrees of freedom here. All right, so degrees of freedom in the numerator is easy. We have columns, and they just go across. And we've got starting from 1, 2, 3, 4, 5, 6, 7, 8. All right, when you look in your table packet, the F distribution actually goes over 4 pages. All right, so there's 2 pages for differences in the numerator. So the next page starts at 9 and it has the same values for the denominator. All right, and then you'll go to the third page and it's going to have the same degrees of freedom in the numerator as this page does, but it's going to go to the bigger numbers for degrees of freedom in the denominator. So it's kind of like you have these 4 pages of the table, and if you set them next to one another and then on, one above and the other below, and you lay them out, you could continuously see how the degrees of freedom numerator are split over 2 pages, and the same thing with denominator. Okay. So we have for each degree of freedom in the numerator, we have to associate it with several values of for the degrees of freedom in the denominator, because we want to give the F distribution critical values or a range of p-values. Right, so we're going to have a cluster of possible p-values, only going from 0.001 to 0.1, so a little limited compared to some of our other tables. So if we have 2 degrees of freedom in the numerator, and we have 5 degrees of freedom in the denominator, we're going to come over here to this area on the table, and we see that we have this range of values of F. And where we have a smaller value of F, it's going to be associated with a larger p-value, right? And where we have a larger value of F, it's going to be associated with a smaller p-value. And we can see that just like the chi-square distribution. This is what the F distribution is going to look like. The values in our table are, again, those F star critical values. And the probability P is going to be the area in the curve under the distribution to the right of the F. All right, so we don't have a big range of numbers for each of the P's, so we're often going to find when we're working out problems by hand and using this table that our value of F is going to be in between 2 of these values. Right? So then we'll just say our P is in between 2 values that we have. All right, so this table's a little bit tricky when you first start using it, and it's really useful to notice that each of these batches or groups of the p-values are shown in with different shading alternating from one set of p-values to the next. so that you can help you get oriented to where you're looking on the table. All right, so for example, we might have an experiment where we have three groups, like I was talking about before, a control and two different treatments. So in this case, our capital I, our number of groups, is going to be 3. And suppose each group has 10 individuals. So little n sub 1 is 10, little n sub 2 is 10, little n sub 3 is 10. So that makes our capital N the sum of all of those little n's, and the capital N is going to be 30. All right, so let's do the degrees of freedom for this experiment, because you can always do that just based on the description of the experimental design, how many groups and how many individuals there are. All right, so our capital I is 3. Our degrees of freedom between groups is going to be this capital I minus 1. So within groups, that's going to be capital N minus I. So we're going to have 30 minus 3, and that gives us 27. And the degrees of freedom total is going to be capital N minus 1. All right, so 30 minus 1 is 29. And you can see that 27 plus 2 actually does add up to 29 here. All right, so I'm going to have you fill in ANOVA tables from reading the experimental design, and then I will provide you with some of this information. Okay, so I can give you 2 of the sums of squares, and then you can find the other one, because if these 2 add up to one another, but you have the total and you're missing the within groups, right? You can subtract the between from the total and you can fill in this one. All right, so look at these numbers. These numbers can get really big. So don't be worried if you have strangely large numbers that you're squares. All right. So once you have the sums of the squares and the degrees of freedom, you can find the mean square, all right, just by dividing across the rows. So we have 91.467. The degrees of freedom for between is 2. So all I have to do is take that 91.467 and divide by 2 to get the mean square, which is 45.733. For the mean square of the within groups, we have a sum of squares of 276.4. Our degrees of freedom is 27. So to fill in this box to the right, we're just going to divide 276.4 by 27, and we get 10.237. All right, now the next thing to find is our F. And in order to do that, we're going to divide the mean square between 45.7 by the mean square within 10.23. All right, so 45 or so divided by 10 is going to be about 4.5. All right, so it comes out to 4.467. And this significance over here, this is the p-value. Now, software gave us this one, but let's look at the table in order to find it and practice using the table. All right, so looking at the F distribution table, we want to go to the correct page with the number of degrees of freedom in the numerator that we need. So here is 2 degrees of freedom in the numerator. And we have to flip to not the first page, I think it's the third page, second or third page, when we go down to larger numbers in the degrees of freedom in the denominator. So looking for 27 degrees of freedom in the denominator, we do not find it in this table. All right, so we're going to use the number that's closest. If we don't have a number that's in our table, we're just going to use the number that's closest. I know that's not entirely satisfying, but again, using the tables is a way that we can quickly and easily look up these numbers for our p-value, and actually using them helps you understand the distributions much better than just getting the number from a software. So bear with me with having to have an estimated p-value. All right. When you do your real work, you'll get all your p-values from software and they'll be exact. All right, so we've got our 2 degrees of freedom in the numerator. So we're in this column, approximately 25 degrees of freedom in the denominator. So we have these values of critical values of f. And we're going to look for our value of f, 4.467 in here. We've got 4.29. So our value is between that, 4.29 and 5.57. So we're going to go across to the p-values and we're going to see which numbers our p-value is in between. So our p-value is in between 0.025 and 0.01. All right, 0.025 and 0.01. All right, let's go back to that F table. So our significance, our p-value is 0.02. If we were using a alpha of 0.05, we would reject the null, right? So we would have had our hypotheses already. So how are we going to go about doing this hypothesis test? We're going to start by making our null hypothesis, which is that all the means are equal, right? The means of all the groups are equal. Mu1 equals mu2 equals mu3. The alternate hypothesis for the overall ANOVA is going to be mu1 does not equal mu2 does not equal mu3. All right, so they're mutually all not equal to one another. They're either all the same or they're all different. All right, and we're going to use our alpha of 0.05 for our initial hypothesis test. All right, so if we accept the null hypothesis, if we'd had a p-value of greater than 0.05, we'd say, aha, all of our means are equal. We're done. All right. In our sample example, we rejected the null. So our means are not all equal. All right. But here's where things get a little complicated with analysis variance, right? If we reject the null, we don't know which of these means are not equal from one another. They could be all not equal to one another, or maybe mu1 and mu2 are so different from one another that they drive the whole rejection of the null hypothesis. All right, so we're not done with our ANOVA analysis if we reject the null. All right, so we have to do what are called follow-up comparisons. And our follow-up comparisons are going to be asking the question, which means are actually different from one another, right? Which of our means are actually different from one another? And we got to compare them all pairwise. So we've got 2 means to compare. So we can do 2 independent samples t-tests between all the pairs of our means, and then see which of those are significant. All right, so we're going to separate out. We're going to have separate little t-tests now for our mu1 equals our mu2 versus they don't equal each other. Our mu2 equals mu3 compared to they don't equal one another, and our mu1 equals our mu3. All right, so if we've got 3 different means, then we're going to have to do these 3 different hypotheses tests. All right, if we have 4 means, we have to do 6 comparison hypothesis tests. And if we have more means than that, it just gets even bigger. All right, so remember that our p-value is a probability, and that the more times that we did an experiment, the more times that we analyzed our data, the more times we're going to get an error, right? So if we set our alpha level at 0.05 and we do a whole bunch of tests using alpha of 0.05, then at some point we're going to reject the null even if it's true, just because of how randomness works. All right, so statisticians feel like when you're testing multiple hypotheses on the same data, you're kind of cheating because you're raising just by doing multiple tests, you're more likely to get that one time that it was an incorrect result or an error. Okay. So the standard correction for this is to have a more strict hypothesis test for these follow-up tests. All right, now there's a bunch of different ways to do this, and if you're using software, then there's much more sophisticated ways to sort of in a nuanced way adjust the alpha level. But this is a really easy and simple way to do that we're going to use for our test in this class. It's very conservative. That means it's going to make it really hard to reject these follow-up null hypotheses. All right, so we've got this alpha level that we started. We did the overall ANOVA hypothesis test with of 0.05. What we're going to do is we're going to adjust that alpha level. And a statistician called Bonferroni came up with this strategy. So it's called the Bonferroni adjusted alpha. And I'm going to write it alpha sub b so that we can tell the difference between it and our original alpha. And we're going to use the total number of tests that we're doing as the way that we adjust the alpha level. Right? We want to make the tests more strict or more conservative. So we want to have a smaller alpha to compare our p-values to. Right. And so we're just going to count up the number of different comparisons we have to make. So if you only have 3 means, you're always going to have 3 follow-up comparisons. So you're always going to be dividing your initial alpha by 3. All right. And that gives us 0.0167. All right. So this is quite a bit more strict. All right. So when we get our p-values from these follow-up t-tests, we're going to compare those p-values to this alpha. Right. This is so conservative that it's possible that we could reject our overall null hypothesis and then not be able to reject any of these separated out pairwise comparisons. Right. So this is not the most favorite of all scientists way to do this adjustment, but that's a simple and straightforward one. It's easy to do. And so that's what we're going to use in our class. So you get the idea of what we're doing. All right, it's a little hard to explain. So this notation, alpha sub b equals alpha divided by number of t-tests. All right, that means how many of these follow-up tests or comparisons you're going to do. All right. And so remember to evaluate your p-values for those follow-up t-tests and only reject the null if the p-value is less than the Bonferroni adjusted alpha level. All right, this isn't going to make too much sense until you see a worked out example, which I have a follow-up video showing, and also when you do some analysis of some data like we're going to do in the lab activity.